Hello, my name is Gabriela Valenzuela, and I'll be presenting my research titled The Quantification of Cannabinol in Hemp Oil by Ultra-High-Performance Liquid Chromatography, Electrospray Ionization Tandem Mass Spectrometry. A little shout out to my research partner Shelby Carlson as she aided in the completion of this project. Now let's get into a little bit of background information on this project. Hemp is a class of cannabis whose main constituents are cannabinoids, and these cannabinoids are being explored as potential therapeutic targets due to their affinity for certain receptors and ligands in our endocannabinoid system. Cannabinol, or CBN, is our main focus of the study as it possesses immunosuppressive properties and can be an aid for those who suffer from chronic pain. Now, quantification of these products due to their medicinal uses is important in order to meet medicinally regulatory requirements. Therefore, we have developed a reliable and accurate method in order to quantify CBN among 12 cannabinoids in hemp oil. The chemical structures of the 12 cannabinoids focused on in this study can be seen on the right-hand side of the screen. The first step in this project was to prepare a 25 milligram per milliliter mixture of hemp oil and methanol in order to recover CBN. The mixture was ultrasonicated for 20 minutes, centrifuged for 10 minutes at 3,000 revolutions per minute, and then filtered. The hemp oil solution was serially diluted with methanol to a concentration of 250 microgram per milliliter. As we will be using internal standard calibration, the 250 microgram per milliliter hemp oil solution was then mixed in a 1-to-1 ratio with an internal standard, in this case a deuterated version of CBN. Thus, the final concentration of solution was 125 microgram per milliliter hemp oil and 0.5 microgram per milliliter CBN-D3. An Agilent 1260 Infinity 2 LC system was employed with the set parameters shown in Table 1. Chromatographic separation was carried out using a phenomenic C18 column. Additionally, isocratic elution was utilized with mobile phases being 2 millimolar ammonium formate spiked with 0.011% formic acid and 73% acetonitrile. The importance of the formic acid was to ensure that our acidic cannabinoids would not co-elute with our neutral cannabinoids in order to meet method validation requirements. Detection of CBN and subsequent quantification of the cannabinoid was carried out using an Agilent 6545 quadrupole time-of-flight mass spectrometer operating in the positive ion mode utilizing an optimized fragmenter voltage of 120 volts. The list of optimized parameters can be seen in Table 2. Chromatographic separation of all 12 cannabinoids was achieved using optimized conditions and can be seen in Figure 2. The next step was to identify and confirm the presence of CBN. First, let's look at the parameters listed in Table 3, specifically the precursor, quantifier, and qualifier ion, as all these ions aid in the confirmation of identity for CBN. Now, the precursor, or the parent ion, is selected in the first stage and is then fragmented to produce many other ions, which includes a quantifier ion and a qualifier ion. The precursor ion is located on the far right-hand side of each spectra in Figure 4. The quantifier ion is going to be your most abundant ion in the spectrum and is used for quantification purposes along with confirmation of identity. The qualifier is your lesser abundant ion, but it is monitored in order to aid in proper identification of our analyte, in this case CBN. Now, all three of these ions are present in our spectra with mass accuracy, so therefore identity and confirmation of the analyte has been achieved, and this allows for proper sample analysis. Analysis of the 125 microgram per milliliter hemp oil extract was then carried out, and its chromatogram can be seen in Figure 5. Quantification of the CBN and the hemp oil extract was achieved using internal standard calibration. A calibration curve consisting of 5 calibrants ranging from 0.01 to 12.5 microgram per milliliter in concentration that had each been spiked with 0.5 microgram per milliliter of internal standard were first prepared. Then triplicate analysis of CBN was carried out on the hemp oil sample, and its measurements were 0.37% with a relative standard deviation of 4.1%. The method is not interfered by any other cannabinoid that may be present in hemp oil due to adequate chromatographic separation achieved using optimized LC conditions, in addition to utilizing a very sensitive and selective detector. The limit of quantitation for our method is determined to be 0.01 microgram per milliliter methanol or 0.008% CBN in hemp oil. In summary, a very accurate and reliable UHPLC-ESI MSMS method was developed, and it was able to quantify the CBN content in hemp oil. This method can assist the United States Department of Agriculture, or USDA, in order to regulate hemp oil products, as the popularity of these products for medicinal uses grow in the U.S. I would like to acknowledge the National Institute of Justice, the National Science Foundation, and the Western Illinois University Research Council in their financial support in the completion of this project.